Hello, everyone. My name is Jack Stahl, and I work in the lab of Dr. Klaus Wahlstedt at the University of Miami Center for Therapeutic Innovation. The title of my presentation is Synergistic Gene Upregulation by RNA-targeting oligonucleotides with different mechanisms of action. Upregulating endogenous gene expression is desirable for a range of human disorders. One such disorder is Dravet syndrome, which is a severe form of epilepsy caused by haploinsufficient expression of the SCN1A gene coding for a voltage-gated sodium channel. Increasing SCN1A mRNA levels improves seizure phenotypes in a mouse model of Dravet syndrome. And there are currently two different oligonucleotide-based approaches being clinically investigated for treatment of Dravet syndrome. These two approaches are antagonats and poison exon excluding oligonucleotides. Shown here are schematics depicting the mechanism of action for both antagonats and poison exon excluding oligonucleotides. So shown on the left, natural antisense transcripts, or NATs, can be transcribed from regions of DNA which are antisense relative to a protein coding region. These NATs exist as an endogenous regulatory mechanism to repress protein production. Therefore, inhibiting the NATs using oligonucleotides, referred to as antagonats, can increase translatable mRNA levels and de-repress protein production. Shown on the right, poison exons are regions of pre-mRNA which trigger transcripts to undergo nonsense-mediated decay. Oligonucleotides preventing the inclusion of poison exons into mature mRNA can increase the amount of translatable or productive mRNA. Using a range of antagonat concentrations, we were able to upregulate SCN1A in SKNAS human neuroblastoma cells by a maximum of 13.4-fold and with an EC50 value of 39.4 nanomolar. Here's a concentration response that's not as pretty, but using a range of splice-modifying oligonucleotide concentrations, we were able to upregulate SCN1A in SKNAS human neuroblastoma cells by a maximum of 3.1-fold with a similar EC50 value of 40.6 nanomolar. In an experiment using 100 nanomolar of SCN1A antagonat in combination with 100 nanomolar of SCN1A splice-modifying oligo, we were able to upregulate SCN1A by 12.6-fold with 100 nanomolar of the antagonat, 2.6-fold with 100 nanomolar of the splice-modifying oligo, and 37.8-fold with 100 nanomolar of both in combination. Here, we use two different oligonucleotide-based gene upregulation strategies, which are being pursued clinically, and show a synergistic rather than additive effect when combined. Our data support further exploring the use of combinations in oligonucleotide-based therapeutics. Synergistic combinations can be explored for other oligonucleotide therapeutics which increase mRNA abundance, such as small activating RNAs, enhancer RNA-targeted oligos, oligos targeting imprinting, and microRNA-targeted oligos. Thank you for listening, and if you have any questions, please feel free to reach out to me via email.